Welcome to the Veterinary Research Channel and to our discussion on cancer chemotherapy. This playlist provides a foundational overview of how chemotherapy works, the complexities of its administration, managing side effects, and the various types of drugs used. We'll explore everything from the basic science of how these drugs target cancer cells to practical considerations like dosing and safety. We'll also touch upon key terminology, discuss how we manage toxicities, look at mechanisms of drug resistance, and delve into the specifics of several important drug classes. Finally, we'll consider prognostic evaluation and future directions in this field. Let's start with a fundamental principle. How do these drugs work?
Absolutely. The fundamental basis of anti-cancer drug activity is the targeting of dividing cells. This is achieved by interfering with processes essential for progression through the cell cycle. The key phases targeted include the S phase, which is dedicated to DNA replication, and the M phase, where cell division, mitosis, occurs. Beyond these phases, a newer class of drugs, signal transduction inhibitors, works differently. They interfere with the signaling pathways that trigger cells to enter the cell cycle in the first place and continue proliferating. Focusing on the traditional cytotoxic drugs, a primary target is DNA synthesis, a complex process. This involves the anabolic creation of the necessary purine and pyrimidine nucleotide triphosphates required for replication. It also includes the unwinding of the template DNA so the replication machinery can access it, and the crucial, high-fidelity process of creating complementary DNA strands. Anti-cancer drugs intervene at all these levels of DNA synthesis. For instance, antimetabolites inhibit the anabolic processes needed to provide the nucleotide building blocks. Topoisomerase inhibitors interfere with the enzymatic unwinding of DNA. Cross-linking agents, through interactions within or between DNA strands, block the processes of strand separation and template processing. Alkylating agents interfere with the replication machinery itself via multiple mechanisms, altering binding and base recognition. The ultimate consequences of these interactions with DNA replication processes can include the generation of DNA strand breaks, incomplete replication, and the triggering of apoptotic signaling, leading ultimately to cell death. Cellular response can manifest in various ways, including mitotic catastrophe, apoptosis, necrosis, autophagy, or cellular stasis. This response depends on a complex interplay of survival and death signaling pathways and is often specific to the particular agent, the dose reaching the critical target, and the cell lineage. Alterations in pro-apoptotic and anti-apoptotic signaling are known to play a role in both tumor development and response to therapy.
And don't forget to like, share, and subscribe, as 50% of all proceeds generated from this channel will be donated towards animal care. Thank you, and as always, keeps those minds inspired, hearts light, and tails wagging.